About some of the work we're doing in cancer single-cell analysis. I'm going to present some unpublished data, so I ask that you don't take any snaps of the data slides. So, I think in a lot of ways we've studied tumors as homogeneous masses for many years, and that's got us a long way in our understanding of tumor genomes. But I think in more recent times we've come to realize, of course, that tumors are complex ecosystems of many different cell types, and it's the net interactions of these cell types that govern the etiology of disease, its response to therapy, its metastatic potential, and so on. And so my lab's been very interested in this area for many years, often struggling with technology to gain insights into it. And so we are very kind of pleased with the potential that single-cell sequencing gives us in understanding kind of heterotypic cell interactions and determinants of heterogeneity within the tumor microenvironment. So we kind of recognized this possibility about 5 years ago, and so started setting up a fresh tissue collection program for clinical cancers around Sydney, where there was an opening, an opportunity. We're collecting at every stage of disease progression, so from surgery or patients undergoing neoadjuvant treatment prior to surgery. When patients relapse with metastatic biopsies or go on to clinical trials, we get research biopsies from them. And I also run a national rapid autopsy program at multiple sites around Australia to sample disease at kind of end stage in patients. Our work is being kind of led by our work in prostate cancer, but we have programs in breast cancer, but we have programs in prostate cancer and smaller levels of activity in melanoma and neuroblastoma. So what I want to do today is talk mostly about the kind of technology and approaches we're taking, rather than necessarily about the biological stories that we're pursuing. So thinking about how to kind of develop a biobank that is compatible with single-cell methods, I think this has been very nicely introduced earlier by Al. We wanted to try to address the question, how should we actually be storing our samples? And there are multiple ways to do this. The two most common ways are either analyzing the tissue fresh or using frozen nuclei. Fresh tissue, I think, probably gives you kind of the strongest RNA signal from those cells, but obviously this is very logistically difficult. There's only so many times you can make your students come in at midnight to do the single-cell captures, right? There are other problems too. So in certain cases, you want to be able to triage samples, for example. When you biopsy prostate cancer, often your biopsy doesn't contain any cancer, and so you ideally want to be able to do pathological and other triage steps before you commit to single-cell sequencing. Obviously, you can't multiplex fresh captures as well, or it's difficult to do. There are a lot of advantages to frozen nuclei methods, but in certain settings, for example, in cancers where you have a lot of necrotic cells and low-quality cells, you're not able to remove them prior to capture. And so we were most interested in pursuing a viable cryopreservation approach because we thought it gave us many of the kind of benefits of both of these approaches. But of course, there wasn't really data on the impact on the data that you get from human tissues. So very briefly, I just kind of skim across some experiments we've been doing. Happy to discuss these in more detail later, but essentially we've been taking breast cancers, prostate cancers, and melanomas from patients. When we have a large enough tumor, we dice this into 1 millimeter cubes, and then we either dissociate, capture it fresh, or take some of that dissociate and cryopreserve it in a serum DMSO media. And we compare that to cryopreservation as tissue fragments. And one of the reasons we wanted to do this is that this method is compatible with a hospital pathology department. So we're most likely to be able to get this method to work routinely. So just to show you some of the metrics that we're seeing here, I'm exemplifying it using 3 breast cancer patients here that are clustered. They're labeled based on the processing that's been conducted on those tissues. And really the take-home message here is that comparing fresh to cryopreserved cell suspensions or cryopreserved tissue fragments, we don't see any major changes in the cell populations that we detect at the end. When we look at some of the other metrics that we can examine, so for example, the average number of genes or the average number of UMIs detected per cell, we see that viably frozen cell suspensions have the same kind of data density as freshly captured cells. But you can see in this third box here that in certain cases we see a modest reduction in number of genes and UMIs when we cryopreserve tissue fragments, something like a 20% reduction. And when we look on the bulk level at least, there's very strong correlation between any one of these models with any of the other ones. So moving forward, we've kind of committed then to using cryopreservation of tissue as the core of what we do in our clinical collections. And so what I want to show you now is just the first snapshot of what we call our Breast Cancer Cell Atlas program. So using 250 early breast cancers that we've cryopreserved as tissue fragments. One of the advantages of this bank is this is a single lab bank. It's all done in my lab. Actually, a single technician prepared all the tissues, and it came from a small number of sites. So we hope that that's going to at least help us manage some of the batch-to-batch variability that we might otherwise see. So we're aiming to capture something in the region of about a paltry 1.5 million cells compared to the billion cells we just heard about, to really understand cell types and cell states. We want to learn as much as we can about each one of these cells so that we can go deeper than just gene expression features, and I'll show you a bit of that data in a moment. We're developing a somatic genomics program around these cases so that we can help us stratify and understand the drivers of disease in these patients. Obviously, spatial data is going to be really critical, and I'll show you just a little bit of our first steps into that area. And then I think what's most important is linking it into the clinical and histopathological features of these patients, because ultimately we want to relate cellular or molecular features we find to clinical features that we can observe, right? As an aside, it's worth saying that all of these cases have also been xenografted into immunodeficient mice, and so about 60 or 70 of these have grown as animal avatars. So this gives us both a matched experimental model where we can test hypotheses, but also the ability of these tumors to grow in a mouse is actually strongly prognostic. So it gives us a surrogate for outcome in these patients, even though we haven't necessarily waited long enough to know whether they're going to relapse or not. So at the core of what we do is single-cell RNA sequencing using the 10x Chromium platform. But because these clinical samples are very precious, we get one crack at them generally. We wanted to learn as much as we can about each of them. So one method that we've adopted is CytSeq that I'm sure most people here are familiar with, but essentially it gives you data that's broadly equivalent to flow cytometry, but instead of labeling antibodies with fluorophores, we label them with polyadenylated barcodes, and we read it out with sequencing rather than with fluorescence. We have also developed a method known as RAGE-seq that lets us do targeted full-length transcriptome sequencing from these very same cells, and I'll show you an example of how we're using that. That was published recently if you're interested. And finally, as I mentioned, tissue-based analysis is going to be very important moving forward. Okay, so the quality of this is a little degraded, but nonetheless, I think I just wanted to show you a very high-level view of this first 26 cases from our early breast cancer cohort. We've selected each of the major subtypes of breast cancer are represented in this. This is something like 160,000 cells, and a subset of these cases have undergone CytSeq analysis. And we're using this really to try to get a high-level view of the taxonomy of breast cancer in the malignant cells, the immune populations, and the stromal populations. So drilling down a little bit now into CytSeq, and one of the reasons why we think CytSeq is really interesting and important to these studies is that it lets us link what we find in single-cell sequencing back to this enormous canon of knowledge in immunology, because essentially the methods are reasonably comparable. They're both antibody-based measures of cell surface epitopes. CytSeq is very sensitive. Often the protein is present at much higher levels than its cognate mRNA, and so unlike the kind of zero-filled matrices that we get in our single-cell RNA sequencing data, the CytSeq data is very rich, and we're often dealing with hundreds of counts per cell rather than a handful. There's no theoretical limit to multiplexing. We don't have problems with spectral overlap here. We're just using barcodes, so we can really potentially use very large CytSeq libraries. And ultimately, when you want to think about this in a clinical context, we need to know whether the protein is present on the surface of a cell to help us drive clinical decision making. It's not enough to infer it from the RNA levels. So this is the panel that we've been working with. It's roughly 150 antibodies now, covering kind of key cell type markers across innate and adaptive immunity. We've also got a large number of checkpoint molecules, particularly all the ones that are undergoing clinical development currently, and then a handful of other markers for cancer and stromal populations. So I'll just show you a little bit of data on how CytSeq is behaving in our hands. This first set here is from PBMCs, which work very nicely with this method. You can see this is clustering based on RNA only, and this is a not atypical kind of result that you might see when you cluster PBMCs based on their RNA data. When we now cluster based on this set of 115 antibodies, you can see this increased resolution that we can start to achieve in defining subsets of immune populations. This also works on tissue. It's more difficult, and it's taken a lot of more effort to optimize CiteSeq, but nonetheless, I can say that CiteSeq does work on tissues given appropriate circumstances. This is a metastatic lymph node from a triple-negative breast cancer patient here. Again, clustering just on the RNA data. This is an earlier CiteSeq panel here, but still you can see now we can go from a single CD4 T cell subpopulation to something like 6 CD4 T cell subpopulations that we're able to resolve using the protein data. There are some other really nice features of CiteSeq data, of course. The first is that we can measure things that aren't actually present in gene expression data. So for example, splice variants, this CD45RA-RO is very important in determining whether a cell has seen antigen or not. We can't extract this from short-read sequencing, but you can see that we get very strong signal using CiteSeq. And also other molecules, for example, some of the checkpoint molecules that are really quite difficult to read out of the RNA data give us very strong protein signal here. So we're quite happy with it. So skipping now to that second method I mentioned, RAGE-seq, which is essentially using nanopore sequencing of 10x chromium full-length libraries and then using the cell barcodes to match the short-read sequencing back to the long-read sequencing. This is a very high throughput method now. We can do this on thousands of cells simultaneously. And this is an example of how we're using it, where from an individual patient, we've biopsied the metastatic lymph node and the draining tumor, and we're able to show using long-read sequencing of T cell receptor and B cell receptor that this patient has clones that are shared between the tumor and the lymph node. So in other words, lymphocytes have trafficked into the tumor. Not only that, we're able to detect expanded clones that have proliferated. And because we're sequencing the full-length lymphocyte receptor, we can gain a lot more information. And so, for example, in the B cell receptor, by sequencing this region of the mRNA that encodes this region of the antibody, we can tell whether these are naive or memory B cells. This information tells us the isotype of the antibody that, again, is very difficult to get from short-read sequencing. And then sequencing of this region of the molecule tells us whether this is membrane-bound receptor or secreted as an antibody. So a lot of information is contained in this full-length sequencing. Excuse me. We're also very interested in the stromal cells. I think we heard from one of the earlier talks a lot of evidence for immune cell interactions with stromal cells. And so we're excited by these subsets we're identifying, particularly these smooth muscle-like cells that resemble a pericyte, but in tissue data we've analyzed, we've actually shown that these cells are disseminated out into the parenchyma of the tumor. And so they've essentially delaminated from blood vessels. Okay, I get it. So I'm very briefly going to touch on some of our tissue analysis. We're using the NanoString DSP platform. One of the problems we have is in the epithelial populations, it's not always obvious what's a malignant cell and a non-malignant cell. And in fact, the ground truth of that is morphology, right? That's the only way we can be really certain currently. So what we're doing is taking signatures of populations that we're uncertain about, using the DSP RNA assays to map them into space and then see how they associate with different morphologies. So here's an example where we've taken cancer regions and then we've actually peeled off the luminal and myoepithelial cells from the entrapped normal ducts, and we can look at expression of this gene, and it shows you that in fact it's a marker of normal luminal cells rather than the malignant luminal cells next door. So this helps us have more confidence here. And so we're now really excited to use the kind of kiloplex approaches where NanoString have very high-plex RNA assays coming through. The last thing we're doing is working with Joachim Lundberg and his student Alma to map our signatures into their spatial transcriptomics data. This is essentially using a probabilistic model to predict the proportion of each of these cell types in each of the dots. And I think the take-home I want to show you here is it's telling us that this subset of CAFs, these inflammatory-like CAFs, have reciprocal localization to these myofibroblastic-like CAFs. So this is we're really excited about the potential to go back and forth between droplet data and spatial data like this. So I think just to summarize, we're building these cancer cell atlases. Viable cryopreservation is a really useful tool for preparing tissue collection, but obviously not perfect, and this is a conversation we need to keep having. These multi-omic methods are teaching us additional things about these cases, and I think spatial analysis is going to be a wonderful addition to this. And I'm really looking forward to meeting as many of you today as possible and talking about new collaborations and team science opportunities. So I just want to thank the people who've done this. This is obviously a large group of people responsible for this work, particularly Sunny, Gami, Kate, and Dan, and our funding bodies, and the clinicians, Jean, Sandra, and Mun, who make it possible for us to analyze these samples. Thank you.